Hi, I'm Hannah. I'm talking about the characterization of substrate diversity in the UDP glycosyltransferase and BHD acyltransferase enzyme families. Um, to start off, plants produce a variety of secondary metabolites that are vital for their interacting with the environment, including processes like defense, pollination, and structural support. To produce these complex molecules, one type of enzymes that plants use are called the transferase. Transferases catalyze the transfer of a moiety from a donor to an acceptor substrate, enabling more complex scaffolds and contributing to chemical diversity. So here we're talking about two enzyme families, the BHD acyltransferases and the UDP glycosyltransferases. One of our kind of topics is that though many of these enzyme activities have been documented, only a small proportion are accessible and publicly available databases. So this poster has two aims. The first is kind of to directly address this, is that we are curating UGT enzyme substrate relationships extracted from the literature for eventual inclusion in public databases like UniProt. And then the second is to modify a BHD structure and measure changes in its substrate specificity, linking structure to function. So starting off with the UGTs transfer a sugar moiety from a UDP conjugated donor to an acceptor. Using a tool developed by our lab called FuncFetch, over 1,000 UGT substrate relationships were extracted from 472 unique papers. Here is demonstrating kind of the results of this. So among the acceptors for the UGTs, those that have an identified superclass, there is a broad diversity of compound types, which kind of highlights just the diversity of substrates that these enzymes can utilize. This also shows that there's so much more diversity in UGT acceptors compared to the donors, which demonstrates the role that transferases play in decorating these complex molecules using much smaller moieties. So moving on to the BHDs transfer an acyl group from a CoA conjugated donor to an acceptor, kind of similar process. An example here is the hydroxycinnamol transferase, or the HCT, which natively uses shikimate as an acceptor and coumaryl CoA as a donor. And this is actually a key step in lignin biosynthesis. So HCT is known to accept other substrates, which leads us to ask the question, what determines substrate specificity? And we used Arabidopsis thaliana HCT to answer these questions. So our focus was on a flexible loop called an IDR, or an intrinsically disordered region, that overlaps the acceptor binding site in BHD enzymes, which is here highlighted in red. We also used HCT because different clades in the BHD family have different substrate specificities and IDR flexibility, and the HCT clade has more IDR flexibility as well as substrate promiscuity, possibly like referring to a connection there. And so our hypothesis was that reducing IDR flexibility will allow us to modulate substrate specificity. So first we targeted proline amino acids for mutation because comparative studies had revealed that proline and the amino acid sequence encoding for the IDR actually increased IDR flexibility. So we made 4 proline to alanine mutations to increase rigidity in the IDRs. And this is demonstrated here using the IUPRED2A tool, where scores above 0.5 indicate a disordered region. And so you can see that successfully between the wild type and the mutated, we were able to make the IDR more rigid. And to test this mutation on the substrates, we did E. coli transformation, colony PCR, protein expression, and protein purification in order to extract the enzyme. And then we did enzyme assays and liquid chromatography mass spectrometry analysis in order to determine the activity. And this was repeated with, these last two were repeated with different substrates, different acceptor substrates, but they all used the same donor substrate, coumaryl CoA. What we found was that 3 out of 4 substrates, the enzyme activity was not significantly affected by the mutant, though there was a significant decrease in product output when using naringenin as the acceptor substrate. So this kind of hints that there is, we are able to kind of modify substrate specificity, though the effect might not be enough yet. And so altogether, this poster kind of highlights the great diversity of enzyme activity and also how we're able to control that diversity by engineering enzyme structure. For the UGTs, organizing enzyme substrate relationships can help us create a comprehensive UGT database that researchers can use to predict an enzyme activity from like sequence. And we also showed that substrate specificity can be engineered, though in this case, the number of prolines may not have been sufficient. So next steps should be more proline to alanine mutations in order to further tune that substrate preference.